Hi everyone, let's continue our discussion of the development of the quasi-species model where we are incorporating selection, reproduction and mutation into account and in this mathematical framework. So, let's try to think about this. We talked about mutation matrix Q which is a n cross n matrix and Qij represents the probability that when ith genome replicates, the progeny upon replication belongs to genome J. That's what this represents, and we saw that this Q matrix is a n cross n matrix Q11, Qij, and the last entry is Qnn, and so on and so forth. So, we have this as a structure of the matrix Q. How do we think about this? First thing that I would like you to think about is, what does matrix Q look like if DNA replication was error free, which means that whenever a bacterium is dividing, it's copying its DNA to be passed on to the progeny, the DNA replication machinery, the DNA polymerase does not make any errors whatsoever and it's 100% faithful. Just pause the video for a few seconds, think about it for 30 seconds, that what would this mean in terms of giving us the structure of matrix Q. So, if this was the case, then you have Qij as probability that ith genome upon replication gives me the progeny which belongs to the jth genome. Now, because replication is error free, if I and j are distinct, that means if I is not equal to j, that means these are two distinct genotypes, then the probability associated with this transition is equal to 0, because there is no way my ith genome upon replication is going to give me the jth genome when the DNA replication is error free. On the other hand, if I is equal to j, what that means is the probability of this event happening is equal to 1, because every time a genome replicates, the progeny is passed the exact same genotype as was there in the parent, because DNA replication is error free. Hence, the progeny and the parent are always carrying the same DNA and the probability of this event is equal to 1. And what does this tell us about my mutation matrix? This tells me that the mutation matrix Q, whose elements are Q11, Q12 going all the way up to Q1n. For genotype 2, this will be Q21, Q22 going all the way up to Q2n, and for the last one Qn1, Qn2 going all the way up to Qnn. What this means is that in the first row, all of these terms represent that there was a mutation that happened when DNA was being copied, because the starting genotype and the ending genotype are distinct from each other, but that is not permitted if DNA replication was error free. If this mutation event did not happen, but I also know that the sum of the elements on this row is equal to 1, what that means is that Q11 element is equal to 1 and all other elements in this row are equal to 0. So, there is 0 probability that the replication event led to a genotype which was distinct from the parent genotype, and the probability that the progeny DNA was identical to that of the parent DNA is equal to 1. For the second row, this the first element and third to the nth element are the elements which represent that there was a mutation event that happened upon replication. Again, these are not permitted, and hence all these entries are equal to 0, and the only entry which is non-zero is Q22, whose probability of happening is 1, because every time a genome 2 divides, the progeny will have genome 2, because DNA replication is error free. Hence, the elements here are going to be 0, 1, and then the rest of the row will have just zeros. Similarly, the last one will have all of these n minus 1 entries representing mutations. Hence, all these elements will be 0, and Qnn will be the only non-zero entry in this, and you will get zeros all the way, and the last one will be equal to 1. So, what that means is that my matrix Q, if DNA replication was error free, would just be equal to what is called an identity matrix, which has 1 as the diagonal entries, and all other off diagonal entries are equal to 0. And by diagonal entries, we mean these entries whose row number is equal to the column number. So, this entry, for instance, is called a diagonal entry because this corresponds to row number 1, column number 1. This is Q11 entry, first row, first column. This is a diagonal entry because this is second row, second column, Q22 entry, second row, second column, and so on and so forth, going all the way up to Qnn entry, which is nth row and nth column. So, these are all the diagonal elements. And the off diagonal elements associated with this matrix are all equal to 0 if DNA replication was error free. Alright, so if replication is error free, let us visualize our system for a minute and try and understand if we can intuitively guess as to what will happen to the process as we play this forward. We have n distinct genotypes starting from 1 to n, each growing at its particular growth rate f1 to fn, and mutation events are not being permitted, which means that whenever genotype 1 replicates, the progeny is guaranteed to be of genotype 1. On top of all of this, we have constrained that the total population size does not exceed the carrying capacity of the environment, which is k. In such a scenario, what should happen, what should be the eventual outcome of this population? Again, I would like you to just pause the video for maybe a minute or so, think about this problem and try to come up with an intuitive picture of how the dynamics of a process such as this, where mutations are not permitted, will play out when we have a vector x defining the structure of the population and a vector f defining the fitness associated with each of the genotypes that exist in the population, and mutations are not permitted, what happens. So what should happen here is that because fitnesses of each genotype are distinct, that means fitnesses are unequal among the genotypes, there is bound to be one genotype which has the maximum fitness from among the vector that defines fitnesses of all genotypes. And irrespective of what the starting fraction of that population is, let us imagine that the structure of population is defined by this vector x, which is x1, x2 going all the way up to xn, and the fitness vector is just equal to f1, f2 going all the way up to fn. Let's have an fi here, and an xi here. Let's imagine that of all the n values here, this one corresponds to the maximum value. And this xi is not equal to 0, which means what this implies is that at the beginning of the experiment, there is at least one individual which belongs to genotype I. So, it is not as if there is no individual in the population which belongs to genotype I. Then in that case, this one or more individuals which belong to the ith genotype, which has the maximum fitness rate, then that these individuals are going to grow faster than every other genotype that exists in the population, and eventually selection will ensure that this xi, this genotype outcompetes all other genotypes in the environment, and this eventually xi approaches 1 and all xj's where j is not equal to I approach 0 as time moves forward. So, this is going to be the outcome of the evolutionary dynamics of these n genotypes if mutations are not permitted. So, again this is the scenario when mutations are not permitted, or if another way to say the same thing is if DNA replication was error free. And this here, what this means is you have one genotype going to frequency 1, all other genotypes going to frequency 0, and that one genotype that goes to frequency 1 is the one that corresponds to the maximum fitness. This is an example of sort of survival of the fittest. In this environment. Okay. But let us now generalize this. So, this was a special case in which we were talking that there are no mutations permitted. Let us now generalize this and try and understand that how would this play out in reality when there are mutations taking place, and my mutation matrix that is associated with growth dynamics of these n genotypes is not an identity matrix where only the diagonal entries are equal to 1 and everything off diagonal is equal to 0. But the mutation matrix is far more general in nature and each entry is allowed to be non-zero. So, what happens then? Now we want to generalize this case, and now mutations are permitted, which is a much more realistic representation of what you would expect to happen in nature. If that is the case, Q matrix is much more general and we cannot make any statement about any of the entries. So, all entries could be non-zero. The only thing that we can say about certainty about this Q matrix is that the row sum has to be equal to 1. That is the only thing that we can say, but other than that, and also the fact that every entry on this in this matrix is a positive number, because these represent probabilities and you cannot have negative probabilities of events happening. If an event is impossible, you have probability equal to 0, but not negative probabilities. Alright. So, now when we have this, let us try and understand that how can we write dx1 by dt, which means how does the frequency of genotype 1 change with time. what is the rate of this process happening with time in such an environment. So, to understand this, what we want to see is that if we have genotype, if we have for what is happening to individuals of genotype 1. This is an individual of genotype 1. I am interested in the rate at which this frequency is changing, but what all are the processes which could lead to a progeny which belongs to genotype 1. Let us try and identify that. So, an individual of genotype 1 could be produced if there is another individual of genotype 1 and its replication occurs and there are no mutations. This process would give me a progeny which belongs to genotype 1. On the other hand, I could also have an individual of genotype 2, and when this replicates, mutation occurred with probability q to 1, that the second genotype replicated and the progeny belonged to the first genotype, and that could also give me a genotype 1. This goes on and if I have genotype I, when this replicates, it could give me a genotype 1 with the probability qi1, that genotype I replicates and I get the progeny of genotype 1. So, these are all the physical events, the division events that could give me a progeny which belong to genotype 1. So, how do we now our job is, so first of all you should identify that how many arrows or how many such distinct processes are there which would give me a genotype, which could give me a progeny which belongs to genotype 1. There are n such distinct type of division events which give a progeny 1, and now our job is going to be to come up with an expression for each one of these n events, and that gives me an estimate of at what rate is the frequency of genotype 1s changing with time. So, let us try and write those expressions down. So, dx1 by dt, the first one let us try to write down the expression for this event, that you have progeny of genotype 1 replicating, no mutations happening, I am sorry, the parent of genotype 1 replicating, no mutations happening, and the progeny also belongs to genotype 1. If that is the case, then this expression can be written as x1 f1. This represents the rate of division of genotype 1, which also takes into account what is the current frequency of individuals belonging to genotype 1 in the environment, and multiply that with q11, which is the probability that should this replication event happen, the progeny also belongs to genotype 1. So, this gives me a quantitative estimate of how many genotype 1s are being generated via this arrow. Let us now try to do, let us now try and now do the same for the second one, and which gives me x2 f2. This is the division events that are happening per time for the individuals which belong to genotype 2, but I am only interested in those events which give rise to a progeny which belongs to genotype 1, hence this will be multiplied by q21. And going all the way, the ith expression is going to be xi fi qi1, and there will be n terms in this, and the final term will be xn fn qin. This gives me an expression for all the events which give to a progeny which is of genotype 1. But remember, this setup has been done in an environment where the total number of individuals is constrained to be equal to the carrying capacity of the environment. Hence, there has to be a dilution terms which accounts for individuals being eliminated such that the total population size is constant, and that is where the variable phi comes back into picture. And we have done this derivation in the earlier parts of the class. So, this expression, in addition to all the generation terms, we will have phi times x1 as the rate of elimination of individuals which belong to genotype 1, where phi is the mean fitness. Mean fitness taking into account all the individuals which are present in the environment at this particular time, and this we have already said is x1 f1 plus x2 f2 going all the way up to xn fn. The first thing that you should note here is that this equation is a nonlinear equation. My variables, the f's are fixed. So, let me just write that down. These all the fi's are constant, because for every particular genotype the fitness rate or the growth rate is fixed, it does not change with time. The mutation matrix Q is fixed once I define it. Once I say that that genotype I can change to genotype J with a probability 0.1, that mutation that probability does not change with time. So, these two are constant. The only variables in this setup are my xi's, which are the frequencies of every genotype. These are my variables. If you look at the first n terms, each one of those terms is linear with respect to xi. There is only one of the xi's that comes in each term, and the power associated with that x is 1, hence these are linear. But because of this term which is there to ensure that the population size remains constant, there is non-linearity in this system, because phi contains an x1, there is also an x1. So, there is an x1 square term, then the second term in phi is x2, that multiplies with x1. So, there is a x1 x2 term. So, this makes the equation non-linear, which makes solving this equation a little non-trivial. Alright. So, it is a non-linear differential equation, and of course, we will have one such equation for every genotype that we are talking about. So, we are talking about solving n nonlinear differential equations simultaneously to track the dynamics of this process. So, this equation can be simplified a little bit, and we can write this as dxi by dt is equal to sigma x j f j q j I, j varies from 1 to n minus phi times x I. I leave that to you to sort of try and convince yourselves that if I were to plug in I equal to 1, I get back this equation. If I were to plug in I equal to 2, I would get the corresponding equation for rate of generation, rate of change of frequency associated with genotype 2, and so on and so forth. This is the eventual equation that we have for such a system where we have n genotypes which are coexisting with each other. Now, the question is that what happens at steady state is what we are interested in. So, what we want to find out is what happens at steady state. And when we discussed the simpler of these two cases where there were no mutations being permitted, it was easy to see that the structure of the population moves in a direction such that the fittest genotype eliminates all the other genotypes that exist in the population, and the population is constrained to that particular genotype, and it represented a case of survival of the fittest. Would that same thing apply here as well? To understand that, and these are nonlinear equations whose solution is not so trivial, so we are going to try and use our intuition a little bit to try and understand, can we figure out that what happens at steady state when we are talking of these systems. So, let us draw a fitness landscape where these n sequences are represented. So, we have these sequences or genotypes, and these are the corresponding fitnesses of each of the n genotypes. And now let me say that these look something like this. And you could acquire a mutation and move in a particular direction, and so on and so forth. So, increase in fitness means that you are a genotype which is growing at a faster rate, and this is the fittest genotype, which is the ith genotype associated from the N that I am talking about here. So, if selection were acting by itself, we would have the entire population move and all N individuals will come to this particular genotype. So, all individuals move to this particular genotype if selection were acting just by itself. But what is happening is that there are mutations happening in the system, and at any particular time, if you have N individuals and mutation events also taking place, you would have this genotype mutating into some of the other genotypes. The other genotypes also mutate back into the nth genotype, that is true, but because there are no individuals to begin with at this particular genotype, there are no individuals which are mutating into the nth genotype. Similarly, individuals here could mutate into this, but because selection has ensured that there are no genotypes at this particular sequence, this arrow does not contribute towards any mutational events. So, if the entire population was constrained at the top of this fitness landscape, then you would have mutational events which would diversify, which would create diversity and lead to other genotypes which are distinct from the fittest genotype that we have around there. But once you have these genotypes being produced, these now selection takes place, and these individuals get eliminated because of selection, and you again have individuals belonging to this particular sequence which are being selected for. So, what is happening is that mutation is aiding diversity, because individuals here are mutating into other genotypes. Mutation is trying to create more genotypes in the environment, but at the same time selection wants to get rid of diversity and only select for the fittest individuals that are present in the environment, which in this case correspond to those of sequence I. So, eventually at the steady state of this process, the balance between mutation and selection plays out, and you get a spread of individuals about the mean, where the maximum number of individuals are present at the fitness peak, but in the neighboring genotypes such as this and this, you have a non-zero number of individuals being present, and that plays out because of this balance that we are talking about between mutation and selection. If there was only mutation, you would have every possible genotype exist in the population. It was only selection, you would only have the fittest genotype survive in the population. But because both coexist, there is a fight between the two, and eventually an equilibrium is established where you have some spread about the population, about the peak on the fitness landscape, and that represents the steady state frequencies associated with the population of these N genotypes. So, we will continue this discussion and its implications on movement of populations on landscapes in our next lecture. Let us stop for this lecture. Thank you.